Hi all, welcome back. The last part of the blood lecture that I want to talk about is blood groups and kind of how transfusions and kind of the blood system works. We'll dive more into immunity and how the immune system works. Part of that we'll touch on in this section because of um, when blood is transfused from one individual to the next and it's a different blood type. We'll talk about what happens during that period of time. So first off, in roughly 1910, although his work started in the early 1900s, Carl Landsterner discovered the ABO blood system and the Rh group that's associated with later on, and he won the Nobel Prize for that. At any rate, we've known about ABO blood antigen genes and system for a very long time. The genetics was worked out much later than the 1900s, but the actual proteins or antigens that are associated with the blood was discovered in the early 1900s. Okay, so blood type is really just referring to the proteins that occur on the surface of the blood cells. So these are called antigens. Okay? And so the antigen will determine what reaction you might have. And what determines what that antigen or what that protein is your genetic code. We talked about this many times. You talked about this in lots of other classes. Your proteins that are being generated by your body are the result of your DNA, of your genetic material. So if you have a different protein on the outside of your red blood cell than someone else, it means that your genetics is different. And so that's where we get different blood types from. Okay, so that knowledge of kind of what antigens is present on the red blood cell is really what allows us and for the most part is very meaningful for things like transfusions. And this happens a lot in healthcare system. We need to know the blood type of the donor and the recipient, because if you transfer blood that doesn't have the same antigens, depending on the blood type, they may have antibodies against that antigen. Okay, and we'll come back and we'll look at this. If they do have antibodies against that antigen, you can get what's called agglutination. Okay, it's an immune response where it takes and it grabs everything with that antigen. These antibodies grabs everything with that antigen and it just combines them all together into a very large clump. And if that happens in your red blood cells, it's like forming a clot. And those red blood cells are now basically useless and they can't move through capillaries. They have a hard time moving through veins. They can clog up regions and you can have major complications. So let's look at that antigen antibody system. So an antigen is any molecule that can invoke an immune response. Okay? Sometimes these are molecules that are floating around in the plasma. Sometimes these are molecules that are stuck to the outside of a cell. Right? Like sometimes these are molecules that you have a eosinophil that just destroyed a trypanosoma, just engulfed it, brought it in, destroyed it, and then it's releasing all this material from the destroyed trypanosoma back into solution, and you got all these little proteins and lipids and carbohydrates. Those all could be antigens. Okay, so it's really any foreign material or self-made material that can trigger kind of a response, an immune response. Excuse me. Antibodies, on the other hand, is what's going to react to the antigen. Okay, and they do so by triggering lots of different immune responses. They could trigger agglutination. Okay, so the combining, they could trigger phagocytosis. They can trigger the complement system. There's lots of ways at which these antibodies can react to the antigen. Okay? In the case when we're talking about blood transfusions, we're talking about incompatibility. We're talking about one individual with an antigen on the outside of their blood, and that's being donated to another individual which has an antibody against it. Okay, and that's where the agglutination or the agglutinated red blood cells will come into play. So agglutination is the clumping of red blood cells, and it occurs when antibodies are reacting to the antigen on a donor cell. Excuse me. So in the most part, when we're talking about transfusion issues, we're talking about ABO or Rh group issues. Now, if you remember back from general biology, you might remember that the outside of all your membranes, even your red blood cells, are just covered in proteins. There's lots of different proteins. There's proteins that allow things to come in. There's proteins that allow things to exit. There's proteins that stimulate cell-cell communication, all kinds of things. But the ones that we're most concerned with from an agglutination point of view is the ABO and the Rh groups. Okay? And you can have lots of issues with transfusion. You can get things that are going on. You could form jaundice, which means that your liver's not working correctly. Could be due to a clotting of red blood cells that are making it to the liver and doesn't allow. Severe pain can occur because you might form a clot and then the tissue on the other end of that clot isn't getting the oxygen, the nutrients it needs to survive, so it starts to deteriorate. Headaches, all kinds of things that can go on. And we may talk about some of these a little bit, but it's better just to transfuse the right blood type than the wrong blood type. Excuse me. So the compatibility, really, we're looking at two main antigens. We're looking at antigen A and antigen B. Okay? And so those are the two antigens that are occurring on the outside of the red blood cells that we're most concerned with, whether they're present or absent. So antigen A and B are actually carbohydrates which are attached to a glucolipid. So we have proteins that are bound to lipids, okay, that have carbohydrates attached to them. Okay, so remember, it's not always just a protein. Sometimes there's a few of these biomolecules that are linked together, and that's what it happens to be. But regardless of that, that's what the antigen is, and the antibody is going to react to that. So transfer compatibilities, of course, this should make sense to you. We've talked about this in lab and things like that. You just need to make sure that the antibody, so what's going to react to the antigen, is not present in the individual that is becoming the recipient of the blood. Okay? And so you have to ensure that that antibody is not going to be present. So, for example, if you, pardon me, if you have a type A blood, it means you're producing A antigens. If you have type B blood, it means you're producing B antigens. A blood, A antigens, but A blood, B antibodies. B blood, B antigens, but B blood, A antibodies. So you can't take blood from a person with A blood type and give it to a person with B blood type because they have the antibodies against the antigen that's present. So let's look at frequency of the different blood types, and then we'll get into kind of what does it mean to have O type, A type, B type, etc. Okay, so for the most part, O is kind of the most prevalent in most groups of people in the world. And then A or B, but AB is the least common for all populations. And so for the most part, it shouldn't be a big surprise. Native Americans and Hispanics, which these two groups are directly related to each other, that their blood types are very common in numbers, or that they have a high frequency of type O blood type. And so other individuals that might be fairly related to each other, Chinese, Japanese, Korean individuals, they have very similar blood types, and so individuals who evolved from similar populations will have similar blood type. That shouldn't be much of a surprise, and that's what we see in the blood typing. So let's look at just the 4 blood types and what does it mean. So again, like I said before, if you have type A blood, you have anti-B or you have B antibodies. Okay, so you have all these little antibodies, which are immune functions, okay, floating around in the plasma, okay, that can attach to the antigen B. Okay, so this blood type is fine because these antibodies aren't going to attach to it. They only attach to B antigens. Okay? B blood type, they produce B antigens, but antibodies against A. Okay? So not compatible. Stick this in here. These antibodies are going to attach to it. Okay, and bad news. Stick this in here. These antibodies are going to attach to it. Bad news. AB, look, no antibodies, none. So their immune system doesn't react to either antigen, neither A or B. Okay? And then in the case of O, both antibodies, but no antigens. Okay, so yeah, they don't have any antigens, so you can put them wherever you want, okay? But you can't put whatever you want back in. Okay, because they have antibodies against both. Okay, all right. So agglutination, okay, is when you are sticking say a blood type in that has an antibody against it. So in this case, we're putting A blood with B blood, okay? And or we could say we could put A with O blood. It doesn't really matter because they also have anti-A antibodies. And when that occurs, these antibodies will attach to those antigens. And they just keep attaching to all the antigens they can, and they form these large clots or these large components of red blood cells, all grouped together in large kind of clots like this. When that occurs, your immune system can take over and will recognize this as kind of a foreign body that needs to be broken down and destroyed. And so your immune system will come down and it'll phagocytize this. It'll eat it. So the macrophages will come in here and just consume all this entire mass, because it has no functionality. It can't work because the surfaces of the red blood cells are not exposed enough. You can't get this into the lungs. It doesn't travel through the veins or the arteries very well, can't make it into the capillaries. So often that will just excite the immune response to start consuming this. Okay? So let's look at universal donor versus recipients. So type O blood, because it lacks antigens, there are no antigens, so there's none of that protein, lipid, carbohydrate component on the outside of O blood. They can donate their blood to anyone, to any blood type. Okay, that makes them a universal donor. Okay, so it doesn't matter if the person has anti-A or anti-B, okay, because there's no antigens, it's a universal donor. Okay. Type AB, on the other hand, is a universal recipient. They don't have antibodies, so they can get blood from A or B or O or AB because there's no antibodies. There's no immune response against the antigens. Okay, and so they are what we call a universal recipient. They can't donate to very many people, to just AB individuals. Excuse me. But they can accept blood from anyone. Okay. All right. Now let's talk about the RH group. Okay. The RH group gets its name from rhesus monkeys because that's when it was first discovered. So yes, all primates have the same blood type system. Okay, so monkeys have a positive RH factor or a negative RH factor. They also have ABO systems. Chimpanzees do. Like all the primates have the same. Now, they might have it in different numbers. Their dominant blood type might not be O. I think in chimpanzees it's B, but don't quote me on it. But nonetheless, they still have ABO system and the RH system, which was discovered in rhesus monkeys by Carl Landsterner. Okay. So, anyways, the RH factor is also important. Okay, so it's not just the antigen that, whether it's a antigen or a B antigen, we also have to look at the RH factor. Okay, so the RH antigen, which would occur. Okay, and so whether you have it or don't have it. Okay, so that would be whether you are RH positive and you have the antigen D, okay, or another type of RH antigen. There are a few different types, but that means you would have an antigen present, an RH antigen, or you're RH negative, which means you do not have an RH antigen that's present on your red blood cells. Okay, so it's just another component. So instead of antigen A, antigen B, now it's RH antigen positive or none. Okay. Okay. Now it is of concern when individuals have the antigen that they will form or that individuals that have the antigen don't form antibodies against the antigen, otherwise they'd attack themselves. But individuals that are negative for the antigen will have the antibodies against the antigen. So individuals that are Rh negative will develop an antibody, or often will develop an antibody against the Rh positive, against the antigen D or some other antigen that's associated with it. Okay. Now, less so than the ABO compatibility, and RH is a little bit less dangerous, but it still can have an effect and eventually become somewhat dangerous as an individual develops or as the days go on, if the RH is miscompatibility. So like if you give someone Rh positive blood, but they're negative, eventually down the road, they will build up antibodies against that positive. Okay. And you can get the similar situation where you have agglutination that occurs, and so you'll form these antibodies, and it'll clump up the red blood cells, and you can get a similar thing that occurs later on. So it's not within, you know, a day or whatever. It could take several days for this antibodies to work. Okay. Now, one of the most serious conditions is not through blood transfusions, but it's more from just normal fetal, normal birthing process. And so what we sometimes find is we have erythroblastosis fetalis, or when you have a fetus that is a different Rh group than the mother. Okay, and that can cause some complications. Um, and in fact, it can mean that the mother's immune system attacks the baby while in the womb. Okay, and so we'll look at it at kind of a picture form of this here in a second. Okay, before we get to that, we can just look at kind of the transfusion types. So if you know your blood type, you might be over here. If you know that, you would know your preferred blood type. It's just identical to whatever your blood type is. So if you're A negative, okay, your preferred is A negative. Just keep it the exact same. That being said, O negative is the universal donor because it's missing the Rh and it's missing the antigens A and B. So it can be donated to every single blood type. So O negative's it. It's universal donor. It can be donated to A negative all the way through O positive. Okay, but the blood type that would be preferred in most cases is whatever blood type you actually are. If you're B negative, it's B negative. Okay, but if you are O negative, you should really think about donating blood because you can be donated to anyone. So if there's no AB positive in the system, in the bank, then you could be donated, or AB negative is a little more rare and a little more complicated. You could be donated to that individual. Okay, so just keep that in mind. All right, so this Rh incompatibility with the womb. So this is on normally the second child. This is what's occurring. Although it could occur after maybe a miscarriage or something like that. So it doesn't have to be the birth of the child, but normally it's the second child. So first child, everything's good. The mother is negative. The child is positive. Okay, but during that pregnancy, that blood from the child is transferred into the mother's system. Okay, the mother does, it filters the child's blood. Okay, so some of that is transferred into the mother's system, and so the mother will get some of that Rh positive blood in her system. And over time, when she's not pregnant, she'll start to build up antibodies against it. Okay, and it will destroy these blood cells. Okay, those antibodies will destroy those blood cells. Those blood cells will never not be in circulation anymore, but the antibodies will be there still. And then if she gets pregnant again, normally this happens like with couples that have multiple children, because the father is what's giving the Rh positive. Okay, so that father produces another child, gives another Rh positive child. Okay, now this child in the womb is Rh positive, but the mother has antibodies against Rh. So when that mother is feeding and taking care of that child in the womb through that blood transfusion back and forth, okay, those antibodies start attacking the child, the fetus, okay, and that can be extremely problematic from a perspective of pregnancy and whether or not the individual is the child's going to make it or not. Okay, so just a couple examples of how Rh might work. All right, when we come back, we'll talk about lymphatic systems and the immune system.